Hello, my name is Andrei Tkatchenko. I am an associate professor of ophthalmic sciences at Columbia University in New York and the founder of Dioptragen Therapeutics. Dioptragen is a startup drug development company which is using its proprietary pharmacogenomics platform to develop novel drugs for the treatment of myopia. It is my pleasure to present Dioptragen Therapeutics, our innovative drug development platform, and our anti-myopia drug pipeline. What is myopia? Myopia is a disease of the refractive state of the eye. It is caused by excessive eye growth leading to a mismatch between the optical power of the eye and its axial lens. In the emmetropic eye, the image is projected directly onto the retina. The hyperopic eye is too small for its optical power, and the image is projected behind the retina. The myopic eye is too large for the optical power of the eye, and the image is projected in front of the retina, resulting in blurred, distant vision. The prevalence of myopia has been growing exponentially over the last few decades. In 1972, the prevalence of myopia in the United States was 25%. In 2000, it reached 42%, and it is projected to increase to 58% of the population by 2050. Similar increases are observed throughout the world. A truly dramatic situation is unfolding in many Asian countries, where the prevalence of myopia in many large cities already exceeds 80%. The overall prevalence of myopia in China is projected to increase from 39% in 2000 to 65% in 2050. Similar situation is expected in Japan. The overall prevalence of myopia in the world is projected to increase to 50% of the population of 5 billion people in 2050. Myopia has already become the leading cause of bilateral visual impairment. 80% of low myopes and 29% of high myopes develop myopic macular degeneration and retinal detachment, and often become blind. In 2050, up to 1 billion people may lose their sight because of myopia. In the United States, up to 200 million people will become myopic, and up to 26 million will become blind. In Europe, these numbers are projected to be even higher. 245 million myopes and 30 million will lose their sight as a consequence of myopia. In China, we will see nearly 1 billion myopic people and 110 million blinded by myopia. India will have 700 million myopes and almost 90 million people blinded by myopia. Even though myopia is quickly becoming a worldwide epidemic threatening to lead to a worldwide health crisis, the treatment options for myopia are mostly limited to optical correction using glasses, contact lenses, or refractive surgery. The problem is that optical correction do not prevent the development of myopia and do not stop the progression of the disease. Neither corrective lenses nor refractive surgery prevent pathological blinding complications of myopia. Experimental treatment options such as ortho-K, multifocal contact lenses, and atropine can indeed slow the progression of myopia, but they cannot stop it. Atropine eye drops have serious side effects. There is an urgent need to develop safe and effective pharmacological options for myopia control. Refractive eye development is controlled by a bidirectional mechanism driven by optical defocus. Negative optical defocus imposed, for example, by a negative lens placed in front of the eye, accelerate eye growth, while positive optical defocus imposed by a positive lens inhibits it. Prolonged exposure to positive optical defocus leads to the development of hyperopia, while excessive exposure to negative defocus leads to the development of myopia. We characterize large portions of the genetic network that controls refractive eye development and identify retinal signaling pathways underlying response to positive and negative optical defocus. We found that negative optical defocus causing myopia and positive optical defocus causing hyperopia activate two different signaling cascades inside the retina. We also found that the genetic network and signaling pathways that control the development of myopia are highly evolutionarily conserved in different vertebrate species as distant as chickens, mice, monkeys, and humans, which means that we can conduct experimental studies and test potential drug candidates in animal models, and the data obtained in animal models would be applicable to humans. We have now generated a large genetic database, which includes information about gene expression underlying response to positive and negative optical defocus, as well as information about gene expression and genetic variations underlying the development of myopia in several animal models of myopia and humans. Using this system's genetics information and a mouse model of myopia developed in our lab, we have developed a pharmacogenomics platform for anti-myopia drug development and testing. This platform uses information about nearly 5,000 genes involved in refractive eye development, information about more than 2,000 drugs and drug compounds, and computational tools to identify potential drug candidates which can suppress the development of myopia. The platform can also identify drug targets for myopia control, which can be pursued for drug development using traditional medicinal chemistry approaches. The identified drug candidates can be quickly screened using a mouse model of myopia and either undergo optimization using medicinal chemistry or used in clinical trials directly. Using this platform, we have identified 138 drug candidates with anti-myopic potential. Using Z-score, which shows how well a particular compound can suppress the pathways underlying the development of myopia and stimulate the pathways underlying the development of hyperopia, we have divided these drug candidates into top 10, top 20, top 40, top 80, and low priority candidates. Our platform also picked up atropine as a potential anti-myopia drug and placed it in the top 20 group. We currently perform computational analysis semi-manually, but our goal is to completely automate our drug development platform using machine learning and artificial intelligence, as well as expand it to other ophthalmic diseases such as glaucoma and retinal degeneration. We then tested the top 6 candidates using a mouse model of myopia. As you can see, we induced approximately 11 diopters of myopia in the control group, while the development of myopia was almost completely suppressed in the groups treated with TR302, SM205, FIP387, FE303, and LE420 compounds. One drug, BE336, suppressed myopia by approximately 50%. All drug compounds were tested systemically, which means animals were fed with medicated food for 3 weeks, and we did not see any serious adverse effects in any of the experimental groups, suggesting that all these drugs have very good safety profiles, much more superior to atropine, the only drug currently used for myopia control. As you might know, atropine is very potent poison, which can only be used topically because of its high toxicity. This is not the case for our drugs, which are well tolerated even if delivered systemically. Importantly, all 6 drug compounds belong to 6 different drug classes, which are not related to atropine. This slide shows a timeline to market for our drug pipeline. 5 out of 6 drug compounds have never been used for ophthalmic applications. For these drugs, we will need to conduct IND-enabling preclinical studies to establish their efficacy and safety profiles. We will also need to develop ophthalmic formulations for these drugs and test them for safety in rabbits. Ideally, these compounds would need to be tested in a primate model of myopia for efficacy and safety before proceeding to phase 1 and phase 2 clinical trials. This may take anywhere from 2 to 3 years. However, we have one drug candidate, LE420, which have been used as a generic drug to treat an ophthalmic disease unrelated to myopia for many years. This drug exists as an ophthalmic topical formulation, has very good efficacy profile, and can be quickly repurposed for the treatment of myopia, especially so because it shows very high efficacy against myopia in our preclinical studies. Considering that we already have abundant phase 1 safety data for this drug, we can take it directly to phase 2 clinical trials. We at Dioptragen believe that eye drops is the best option for myopia control because eye drops are relatively cheap, eye drops are safe, with very little, if any, systemic effects. Eye drops can be self-administered, and eye drops can be used for pediatric patients. However, we are also considering other ophthalmic formulations such as extended drug release modalities and extended drug release combination devices, such as contact lenses coated with our drug compounds. Our pharmacogenomics platform was featured on the cover of Trends in Pharmacological Sciences. We also have 6 pending patent applications for the 6 leading drug compounds listed on this slide. And finally, we are looking for partners and investors who are interested in joining our team to bring our drug pipeline to market and to automate our drug development platform. Thank you very much for your interest, and I am looking forward to hearing from you.